Chapter 13, Section 3, Mutations. What are mutations? Mutations are heritable changes in genetic information. This means that the mutations must be able to pass from parent to offspring. Types of mutations. Now and then cells make mistakes in copying their own DNA, inserting the wrong base or even skipping a base as a strand is put together. These variations are called mutations from the Latin word mutare, meaning to change. Mutations are heritable changes in genetic information. All mutations fall into two basic categories. Those that produce changes in a single gene are known as gene mutations. Those that produce changes in whole chromosomes are known as chromosomal mutations. Mutations that involve changes in one or a few nucleotides are known as point mutations because they occur at a single point in the DNA sequence. They generally occur during replication. If a gene in one cell is altered, the alterations can be passed on to every cell that develops from the original one. Point mutations include substitutions, insertions, and deletions. In a substitution, one base is changed to a different base. Substitutions usually affect no more than a single amino acid, and sometimes they have no effect at all. In the example above, the base cytosine is replaced by the base thiamine, resulting in a change in the messenger RNA codon from CGU, which is arginine, to CAU, which is histidine. However, a change in the last base of the codon from CGU to CGA, for example, would still specify the original amino acid arginine. Insertions and deletions are point mutations in which one base is inserted or removed from the DNA sequence. Insertions and deletions are also called frame shift mutations because they shift the reading frame of the genetic message. Frame shift mutations can change every amino acid that follows the point of the mutation and can alter a protein so much that it is unable to perform its normal function. Chromosomal mutations involve changes in the number or structure of chromosomes. These mutations can change the location of genes on chromosomes and can even change the number of copies of some genes. There are 4 types of chromosomal mutations: deletions, duplications, inversions, and translocations. Many mutations are produced by errors in genetic processes. For example, some point mutations are caused by errors during DNA replication. The cellular machinery that replicates DNA inserts an incorrect base roughly once in every 10 million bases. Small changes in genes can gradually accumulate over time. Stressful environmental conditions can cause some bacteria to increase mutation rates. This can actually be helpful to the organism, since mutations may sometimes give such bacteria new traits, such as the ability to consume a new food source or to resist a poison in the environment. Some mutations arise from mutagens, chemicals or physical agents in the environment. Chemical mutagens include certain pesticides, a few natural plant alkaloids, tobacco smoke, and environmental pollutants. Physical mutagens include some forms of electromagnetic radiation, such as X-rays and ultraviolet light. This slide lists some common mutagens: UV radiation, both natural and from tanning beds; X-rays from medical, dental, and airport screenings; chemical mutations such as cigarette smoke, nitrates and nitrites, preservatives such as in hot dogs and other processed food. Barbecuing creates mutagens in the chemicals in food by altering them. Benzoyl peroxide, which is also considered a mutagen, is a common ingredient in acne products. Certain infectious agents such as human papillomavirus, HPV, which is sexually transmitted, and Heliobacter pylori, a bacteria spread through contaminated foods. All of these have shown to cause mutations in the genetic materials of a cell. The mechanisms that these mutagens use to cause mutations can vary. If these mutagens interact with the DNA, they can produce mutations at a high rate. Some compounds interfere with base pairing, increasing the error rate of the DNA replication. Others weaken the DNA strand, causing breaks and inversions that produce chromosomal mutations. And cells can sometimes repair the damage. But when they cannot, the DNA base sequence change becomes permanent. Sometimes these mutations are harmful, sometimes they're helpful. Most of the time, they do nothing. So how do they affect genes? The effect of mutations on genes vary widely. Some have little or no effect, some produce beneficial variations, and some negatively disrupt gene function. Whether a mutagen is negative or beneficial depends on how its DNA changes relate to the organism's situation. Mutations are often thought of as negative because they disrupt the normal function of genes. However, without mutations, organisms cannot evolve because mutations are the source of genetic variability in a species. Some of the most harmful mutations are those that dramatically change protein structure and gene activity. The defective proteins produced by these mutations can disrupt normal biological activities and result in genetic disorders. Some cancers, for example, are the products of mutations that cause the uncontrolled growth of cells. Sickle cell disease is a disorder associated with changes in the shape of red blood cells. Normal red blood cells are round. Sickle cells appear long and pointed. Sickle cell disease is caused by a point mutation in one of the polypeptides found in hemoglobin, the blood's principal oxygen-carrying protein. Among the symptoms of the disease are anemia, severe pain, frequent infections, and stunted growth. There are some beneficial effects. Some of the variations produced by mutations can be highly advantageous to an organism or species. Mutations often produce proteins with new and altered functions that can be useful to an organism in different or changing environments. For example, mutations have helped insects resist chemical pesticides. Some mutations have enabled more microorganisms to adapt to new chemicals in the environment. Plant and animal breeders often make use of good mutations. For example, when a complete set of chromosomes fail to separate during meiosis, gametes that result can produce triploid, or 3N, or tetraploid, 4N organisms. The conditions in which an organism has extra sets of chromosomes is called polyploidy. Polyploidy plants are often larger and stronger than diploid plants. Important crop plants, including bananas and limes, have been produced this way. Polyploidy also occurs naturally in citrus plants, often through spontaneous mutations.